Hey everyone, welcome to my molecular biochemistry series, and this is the first episode, and we are focusing our attention on the structure of DNA. And DNA may not have been present in the very first cells that existed on Earth, something we often refer to as the protocell. It is there that many molecules were just kind of hanging out inside a vesicle-like membrane, and eventually, in order for cells to continue propagating and surviving, they needed something to regulate and store genetic information so they could produce proteins that would improve their survival, the chances of survival. And evolution decided, in a way, that the most energetically favorable method to do that would be through molecular DNA. And so DNA really drives everything in our cells, and it stores all the information needed to improve survival chances. Now, here we have our DNA strand, poorly drawn, but nonetheless, you get the idea. And you can see that we have nucleotides here, and their molecular structure is such that there is a phosphate group, a sugar molecule, and then the base. If you remove the phosphate group, so you remove this guy, and you're left with just this portion, it's referred to as a nucleoside. Just important terms to understand. So if we magnify or zoom in on our DNA molecule, and we can look at individual atoms, we can see here what it would look like. And the double helix forms in an anti-parallel fashion, such that there is a 5' 3' end parallel to another 5' 3' backbone, but they are read in opposite directions. And the 5' end usually ends with a phosphate group, while the 3' end has a hydroxyl group for new phosphates to attach to. And it's always read 5' to 3'. So that's the backbone. Now if we move even closer into the interior, the bases that exist, there are 5 of them, and they are attached or bonded through hydrogen bonds. That is these white dots here, and that is how they are connected. But how do you identify which base is which? A good way to remember that is I have developed some memory devices and used memory devices I've seen in other resources. And so the first thing I always remember is pure as gold. That means that the A and G represent adenine and guanine, and they are purines. But I can't remember what a purine even is. So I go to the next memory device, and I know that a cut pi means that we are cutting a pi, and pi is just one circle, so it has to be either this one or this one, and that pi is very similar to pyrimidine. And so you have 2 pyrimidines here, and they are represented by one circle. Now, which one is which? Well, I have the next memory device, which is methyl 2. And so you add a methyl group. This one has the extra methyl group. This one does not. This one does not. And 2 has 2 oxygens, so this has 2 oxygens. And T represents thymine, so this must be thiamine. Great. That means this one has to be RC, because it's not adenine or guanine. So we have our T and C just through process of elimination. Now we have our purines over here. Which one is which? Well, we need one that forms 2 hydrogen bonds. That would be this guy. And so it must be, using our last memory device, at Grand Champ, and the T represents 2. So this must be adenine. And that leaves this one being guanine. So there you have a detailed look at the molecular basis of DNA and how to identify the different bases, how the backbone is structured. Also, if you remove this methyl group, you actually have uracil. And so if you add your methyl group to uracil, you get thymine. U and T is what's switched from DNA to RNA. And that is an easy way to remember long term the molecular structure of DNA. And these bases are going to drive which proteins are created, and those proteins drive every function in our cells. And of course, as cells come together, they coalesce to form tissues, and what emerges is complex structures such as the human body. In the next video, we are going to go even deeper into the structure of DNA, where we look at how it sits inside the nucleus and how it sits within the cell itself. But before I do that, I actually want to show you guys a molecular model of what DNA would look like using space-fill atoms, which I think is the best model to use to visualize what is happening. So we see here at labexchange.org a model of the DNA double helix. You can see the various different atoms that would make it up, real life atoms. So gray is going to be carbon, red, oxygen, these purple ones, phosphorus, the white ones, hydrogen. And this is something of what it really would look like if you could go at the size, if you could shrink to the size of an atom. You could also look at it in the form of chains, so the two wraps around, or just the nucleotides, the AGT. Also, you could look at real-life images of what DNA could look like. Here in figure E, we can see at different scales, so 200 micrometers squared, 300 to 2500, you can really see where the DNA is condensed in the darker areas versus the more open area where DNA could be and less condensed. And then here you have a model of a nucleosome, which we haven't gotten into yet, so actually I'll leave it there. And you'll see me break that down in a future video. Until then, I'll see you guys later.